Welcome, geniuses. I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
For this video, I'm going to cover AS topic 3 on enzyme immobilization. So first thing, what is enzyme immobilization? So it's actually a process where an enzyme is actually attached to a matrix that is most likely inert. So the enzyme cannot move, but they can still bind to substrate because the active site is still exposed. So in this case, we are supposed to describe and explain the effect of enzyme immobilization versus when the enzyme is free in the solution. So let's see what is the difference between these two and what is the advantage of enzyme immobilization. So before we start, the syllabus actually requires us to know how enzyme is immobilized using a entrapment method. So in our lab, we will immobilize our enzyme in an alginate, which the texture is like our agar base. And you realize that on this agar base, there's actually pores. And these pores is meant to allow substrate to enter. So as the substrate enters, it will bind to the enzyme, forming an ES complex. And the result will be product is formed, and product will be released from the alginate bed, and it will be separated from the enzyme. There are actually many methods of immobilization, such as adsorption, where the enzyme is actually attached to an inert support material, and with the active site exposed, enzyme substrate complex can still be formed. And there's another method called chemical bonding, where the enzymes are attached or linked to each other using a covalent bond. So the enzymes are not able to move, waiting for the substrate to come and fit in to forming the enzyme substrate complex. So in our syllabus, we are expected to know the methods of enzyme immobilization, and a typical example is preparing the alginate bits. So what we are doing here is we will mix the enzyme that we desire. It could be a pure enzyme, or it could be an enzyme within a bacteria or fungus, and you're going to mix it with a sodium alginate solution, something like your agar solution. So this is your beaker where you're going to add in your enzymes, and you're going to add in your sodium alginate. So the next thing we're going to prepare will be a beaker with calcium chloride solution. So we're going to use a syringe to get the mixture of sodium alginate and enzyme. And using the syringe, we're going to drip the solution in the form of beads into the calcium chloride solution. So when the sodium alginate solution touches the surface or in contact with calcium chloride, you'll realize that the calcium ion displaced by the Na+, forming an insoluble hard beads of calcium alginate. So the enzyme is actually trapped within the calcium alginate beads now. So after you're done preparing the beads, so this is a step 2. We are going to wash the beads, okay? Wash the beads, rinse it in order to remove all the solution that is actually stained on the surface of the beads, such as calcium chloride, sodium alginate, and then we are going to pack all these beads into a column, usually a burette in our lab. So we're going to pack the beads closely. So when the packing is done, that's where we can prepare a beaker to collect the product, and we can start pouring in the substrate. So for example, if I'm going to use an enzyme amylase that is trapped within the bits itself, my substrate will most likely be a starch. So what do I expect to get? So as the starch is trickling down the bits along the column, so the starch will be entering the bit, react with the enzyme, and exit the bit. So if the starch, not all of them are reacted, they can continue with the next bit and subsequently the following bit until towards the end, most likely all the substrate would have been converted into product. So when you think the reaction has completed, you're going to open the clip and release the product. That's where the product is collected. And how would you know if all the substrate is converted into product? So that's where we can carry out some food tests. For example, if it's starch, I can take a bit of sample and carry out iodine solution test. That's where I'll know that if all the substrate is converted into product, my iodine solution will remain yellow. What if you realize that not all your substrate is converted into product? So we can always repeat the experiment by taking the product and pour it through the column again until we are very sure that all the substrate is converted into product. So how is this method better compared to your enzyme free in the solution? So we can see that if you have free enzyme in the solution, for example, you add in your substrate and your enzyme is added in. So after a fixed time, you will expect to see a product is produced. But the thing here is, your product will be mixing with your enzyme. So it's very difficult to actually separate your enzyme from the product. So we're going to look at the advantage of enzyme immobilization versus enzyme free in a solution. So before that, let's look at factors affecting the rate of reaction. So it's the same reason as the factor that affects enzyme activity, such as number one, we have temperature. So obviously, if you provide the enzyme with the optimum temperature, the rate of reaction will be higher. And with the optimum pH, they will also achieve the maximum rate of reaction. The higher the concentration of enzyme, the higher the rate of reaction. And the higher the substrate concentration, the rate of reaction also will increase. However, if you're referring to the technique of enzyme immobilization, you realize that the bead size does affect the rate of reaction. So, which size actually is suitable for enzyme immobilization? This is where it's related to surface area over volume ratio, where the smaller the bits, the greater the surface area over volume ratio. Then you will have higher rate of reaction. And another thing, it could be affected by the column length and the flow rate. So now, let's look at the advantages and disadvantages of enzyme immobilization. So, the advantages of enzyme immobilization will be the disadvantage of free enzyme in solution. So take note of that. So in exam, they like to come up with graphs. So they will actually talk about what is the advantage of enzyme immobilization, or sometimes it could be the advantage of free enzyme in solution. So you just need to know how to flip the points. So let's focus on the advantages of the enzyme that's being immobilized. So number one, we know that this technique can be saving costs because your product is separated from the enzyme. Therefore, you don't need to spend money on purification of product. So always make sure that anytime you talk about saving costs or it's more costly, make sure you actually qualify it by giving an explanation. For next point, we can say that your enzyme will be thermostable and pH stable, means they are not easily affected by the surrounding environment because they are separated from the solution, which is the environment. Number 5, we say that the productivity could be higher as you can keep pouring in your substrate and continuously harvesting your product. So you don't need to waste time in like cleaning up the beaker, refill again the substrate and enzyme. So in this manner, your productivity will be higher. And lastly, the enzyme can be reused, which is also one of the reasons why this technique is more cost saving.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with one or two past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.